Yo, what's up everyone? Kashi here. In today's episode, we're going to be learning about adenylyl cyclase and cAMP. So in the last episode, we looked at GPCRs and G proteins, and we learned that when a messenger molecule comes and binds onto a GPCR, it can cause the G protein inside the cell to split up into two halves, and each of these halves can go and activate different enzymes and produce different responses. Today, we're going to focus all about a specific type of enzyme that is called adenylyl cyclase. We're going to learn about how this adenylyl cyclase is activated and what it does, which is produce cAMP. And we're going to learn about how this produces responses inside the cells. Let's get into it. So before we get into how adenylyl cyclase works, first let's take a look at how this enzyme can get activated and inhibited. So remember before we learned about G proteins, right? There's actually lots of different types of G proteins, and two common types of G proteins are GS proteins and GI proteins. So in GS proteins, the S stands for stimulatory. And as the name suggests, these stimulate adenylyl cyclase, so they increase its activity. Then, in GI proteins, the I stands for inhibitory. So these inhibit adenylyl cyclase, or they lower its activity. So let's say you have your GPCR, right? The GPCR is bound to your cell membrane, and you got a messenger molecule which comes and binds to it, right? Well, on the inside, remember you have your G protein, right? So you got your GDP, and then you got your alpha subunit, your beta subunit, and your gamma subunit. And then when your messenger molecule comes and binds, this GDP is replaced with GTP, which is active. And depending on what the messenger molecule is, what type of messenger molecule you're dealing with, and what type of GPCR you're dealing with, this G protein on the inside that gets activated can be different types. So let's say this messenger molecule and GPCR activates a GS type of G protein. So then this can go over to your adenylyl cyclase and increase response. And let's say over here we have a different type of messenger molecule that goes and binds onto a different type of GPCR. And here, instead of activating a GS G protein, we activate a GI G protein. So this, instead of activating adenylyl cyclase, this goes over and inhibits adenylyl cyclase. So as you can see, these two different types of G proteins are working against each other. One increases response, while the other decreases response. So basically, it strikes a balance, and overall, it modulates the overall adenylyl cyclase activity in the cell. Another thing to note is that both for these GS G proteins and GI G proteins, their responses of increasing or decreasing adenylyl cyclase activity eventually gets terminated by your GTPase enzyme, which reverts GTP back into GDP by dephosphorylating it, so it goes back to its original inactive conformation, and so it terminates the response. So now that we know how these different types of G proteins can control the activity levels of adenylyl cyclase, let's take a look at what this enzyme actually does. Alright, so your adenylyl cyclase is an enzyme found in your cell membrane, and when it gets activated, what it actually does is it transforms ATP into something called cAMP, which stands for cyclic AMP. Cyclic AMP is important because it's what we call a second messenger, and it is essential for producing responses inside the cells. So how does this work? Well, your cAMP or cyclic AMP will go and activate things called kinases. Well, what are kinases? Kinases are enzymes inside the cell that are made of two different parts. You have your regulatory components, your regulatory part, and then the other part is called the catalytic parts. And as the name suggests, catalytic part, this is the part that actually catalyzes reactions. This is the part that's responsible for reactions inside the cell. The regulatory parts, this one up here, is responsible for keeping the catalytic component under control. So it will be attached to the catalytic component and it'll kind of hold it in place. It keeps it controlled. So when your cyclic AMP or your cAMP, right? So it's going to make its way, so you have your, let me just erase that, make it more clear. So you have your cAMP. It's going to make its way over to your regulatory subunit. And here you have little binding sites for it to attach to. So it's going to attach to these binding sites. And what that does is it changes the shape of this regulatory components. And that actually breaks these connections. So now the catalytic component is free. It can leave and it can go and do stuff. It can go and catalyze reactions. So just to review that real quick, the adenylyl cyclase transforms ATP into cAMP, which is what we call a second messenger, and that goes and binds to the regulatory component of kinases, which are enzymes in the cell, which frees the catalytic components to go off and do responses. But how does the catalytic units of the kinases do responses? It activates something called the amplification cascade. Okay, so now that the catalytic unit of your protein kinase is free and active, what it can do is it can make its way over to these enzymes, right? And it can activate them through phosphorylation. So what it does is, like, let's say you have ATPs, right, floating around inside the cell. It can steal a phosphate group from this ATP. So now this ATP becomes ADP because it now has 2 phosphates instead of 3. And it transfers this phosphate over to your enzyme. So now your enzyme has a low phosphate group on it. And it can do this for all of these enzymes. But here's the cool parts. Those enzymes can go and activate more enzymes. So each of these original enzymes can now go and activate each of these enzymes. So we can go and activate all of those. And then each of these enzymes now can go and activate all of these enzymes. So as you can see, the total number of enzymes has increased a lot. You've gone from just one catalytic unit to a few of your first enzyme types to now so many enzymes. And this can continue and continue until you can get like a huge number of enzymes. So what we call this, right, as we go down this pathway, we call this the amplification cascade. Because it's a cascade from your first set of enzymes to your second set of enzymes to your next set of enzymes, the next set of enzymes, and every time we go down, the total number of enzymes increases. So the total response is amplified. So now what all this final big set of enzymes can do, they can do your final response, which is, let's say, turning some sort of substrates into a product. So let's say, I don't know, they want to turn glycogen into glucose. Whatever your final desired response is, now you have this whole huge bank of enzymes working towards to produce this final response. So the cool thing is you've basically converted like one original signal into this huge response through this amplification cascade. That's how responses in cells happen. You have one messenger molecule that comes and binds onto a receptor that can create a much bigger response. You're turning a small signal into a huge response, and it's all due to the help of the amplification cascade, powered by secondary messengers such as cAMP and protein kinases. Alright, so if you guys are feeling confused at this point, don't worry, I fully get it. I was fully lost when I first learned this. Let's go over it nice and easy. Alright, so you have your cell membrane here, right? And along it you have receptors such as GPCRs, and you have enzymes like adenylyl cyclase. And let's say you have a messenger molecule that comes and binds to your GPCR. When it does, then G proteins inside the cell can get activated, such as GS proteins and GIG proteins, which can activate or inhibit adenylyl cyclase. So GS increase ACE activity, GI lowers adenylyl cyclase activity. So together, these modulate and balance overall adenylate cyclase activity. Now, adenylate cyclase, what it does is it turns ATP inside your cell into cAMP, which is a common second messenger that goes over to your protein kinases. And remember, your kinases have a regulatory component which keeps your catalytic component in check. And what cAMP does is it goes and binds to the regulatory components, and then so now the catalytic components, they are free and active, and they can go off and do their thing. And what do? Well, they go off and they go to enzymes in the cell, and they activate these enzymes. So I'm going to do a little asterisk to show that they're active. They activate them by phosphorylating them, and then these enzymes can activate your next wave of enzymes, and then those enzymes can activate your next wave of enzymes. And I'm only going to draw this for conciseness, but this can go on and on and on, and then eventually you get to your final wave of enzymes that produces your desired final response. And that's about it. So it's a long process. It goes from GPCR to G proteins to enzyme like adenylyl cyclase to cAMP second messenger to kinases, which activate some enzymes and activate some more enzymes that activate more enzymes, and then eventually your final response. And that's the whole picture. Alright, so all this time we've been learning about kinases, second messengers, the amplification cascade, and all these things work together in order to produce a desired response inside cells. But when we look at our cell signaling framework, after response must come termination. The desired response that we produce at the end of our amplification cascade, we need to terminate it. We can't let it go on forever out of control, or else it can be dangerous. So how does this happen? There's actually a couple of different termination mechanisms in process. So in order to terminate our responses and prevent them from going out of control, our amplification cascade has three main termination enzymes. Let's take a look at them one by one. First one we got is GTPase, and this is one that we already just looked at. GTPase, basically, it's the GTPase, it terminates G proteins by turning GTP back into GDP. So now, but it does this by dephosphorylating, removing a phosphate group. So now that we're back to the inactive GDP, our stimulatory G proteins can no longer activate adenylyl cyclase. So activity and response goes down. But there are two more types of termination enzymes. You have PDE, or phosphodiesterase, and you have phosphatase. Let's take a look at how these two enzymes work. So the next enzyme we got is phosphodiesterase, and this enzyme is a terminator of cAMP. So phosphodiesterase comes along and it turns cyclic AMP into just AMP. So whereas cAMP has this cyclic component, so it has like a cyclic structure, so maybe something like this, it gets turned into just a non-cyclic structure. And without the cyclic structure, it can no longer act as an effective second messenger and go and activate your kinases, and then it allows your kinases to go and activate your enzymes and allow the amplification cascade to continue. So it's like a domino effect. By getting rid of this important second messenger up here, it inhibits everything that comes afterwards. So that's one enzyme. Now let's look at your third termination enzyme, phosphatase. So your phosphatase enzyme is actually really similar to GTPase in the sense that both of them do dephosphorylation. But whereas your GTPase, it dephosphorylates your GTP, turning it into GDP, your phosphatase instead, it dephosphorylates your active enzymes or proteins. So in the amplification cascade, a lot of times when these enzymes are activated, and those enzymes will go and activate more enzymes, a lot of times they're activated by transferring a phosphate group onto them, so they become phosphorylated. They have this little phosphate group attached, and so they are active. But when phosphatase comes along, this enzyme removes this phosphate group. It causes the phosphate group to leave. And without the phosphate group, the enzyme is no longer active. So all of the, it can't activate any more enzymes. So the phosphatase group inhibits the amplification cascade. And it can act along different enzymes. Maybe it dephosphorylates and deactivates enzyme 1. Maybe it does it to enzyme 2 or enzyme 3. It can act in different areas along the amplification cascade. But the general idea is that it dephosphorylates some sort of active protein or enzyme. And those are your 3 main termination enzymes. So having these 3 different termination enzymes may seem like a bit overkill, but this really emphasizes just how important termination is as a step. Any type of cell response, even one that is important for survival and necessary, it can be really dangerous if it's taken out of control. So termination to keep this response under control is especially important. And a lot of diseases actually work by inhibiting these termination processes. For example, there is a disease called cholera, which inhibits GTPase. So GTPase is not able to turn GTP into GDP and terminate G proteins. And because of this, Adenylyl Cyclase makes an excessive amount of cAMP, which has negative implications. And in a future episode, we'll actually take a look at cholera and figure out why this excess production of cAMP is so dangerous and how we can treat this disease. So, in conclusion, I hope this episode was able to give you a better understanding of how GS and GI proteins work together to control levels of adenylyl cyclase, which produces cAMP, activates protein kinases, which activate enzymes that activate more enzymes, and this goes on and on through the amplification cascade to produce your desired response. And you also have important termination enzymes that keep these processes under control by terminating different paths along this long and complex system. Now, I understand that this is a complex system with a lot of moving parts, and it can definitely be tricky to get a grasp of. What I'd recommend is try to draw as much as you can from memory. Try to draw it out on like a whiteboard, on a big piece of paper, and then refer to this video and fill in any gaps that you may have missed. And doing that a couple of times will really help solidify this process. But if there's anything that you want me to do to help you understand better, if there's anything that you want me to go over, explain again, maybe make some practice problems, please let me know down in the comments. I'm open and willing to hear any suggestions, any tips. I know I can definitely improve. I would really appreciate this. But other than that, thank you guys so much for watching. I really appreciate it, and I hope you enjoyed. Have a great day. Next episode, we're going to take a look at a new type of G protein called GQ proteins and look at their function and how they activate a different type of enzyme. But until then, take care. See y'all next time.